So our speaker for today is Professor Tatiana Besset. Professor Besset obtained her PhD in chemistry in 2009 at Grenoble University with Dr. Green. She then moved to the WWU Münster as the postdoctoral fellow in the group of Professor Glorious. In 2011, she joined the group of Professor Rieck at Amsterdam University as an industry postdoctoral fellow, Eastman Company. From 2012 to 2021, she was a CNRS Associate Researcher Habilitation in 2018 in the Fluorinated Biomolecule Synthesis Group at the laboratory COBRA UMR 6014 Rouen, France, and she was promoted Director of Research CNRS in 2022. Her research career has been recognized with different awards and honors such as ERC Starting Grant 2017, Bronze Medal of the CNRS Young Investigator Award 2018, Jean Pierre Sauvage 2018 Prize awarded by the Division of Organic Chemistry DCO of Société Chimique de France, SCF, and the Time Chemistry Journals Award 2020. In 2022, she was elected as Chemistry Euro Fellow Class 2020-2021 and early 2023, she received the 2023 RSC Fluorine Prize. Her research involves the design of new transformations involving transition metal catalysis by CH bond activation and the development of new strategies in organofluorine chemistry. Thank you so much, Professor Besset, for joining us here, and the stage is all yours now.
I would like to thank you for this amazing opportunity to be there and to be part of this virtual event. I'm very pleased to show you and to talk today about our main contribution in the field of organofluorine chemistry. So my research group have been particularly interested in the design of original tools in organic synthesis. To do so, we like to converge and to merge catalysis and reagent design to tackle synthetic issue in interactive CH bond functionalization as well as in organofluorine chemistry. As you know, the fluorine atom is a very special one. Up to 20 to 25% of pharmaceuticals and up to 40% of agrochemicals contain at least one fluorine atom. So you can find it everywhere. And the unique features of the fluorine, such as its high electronegativity, small size, and the carbon-fluorine bond strength, make it so unique. Indeed, since the late 50s, you can see that an increasing number of study and development towards the synthesis of those fluorinated compounds, from this 5-fluoroacyl and fluorocortisone, for instance, and then reaching in 2019, 43% of the fluorinated molecules approved by FDA contain at least one fluorine atom. However, there are less than 20 natural fluorinated molecules. So there is a key challenge for us as a scientific community and as synthetic organic chemists to develop new tools to tackle persisting challenges in organofluorine chemistry. Here I cite a couple of them. I mean, there is an urgent need to design emergent fluorine-containing moieties in a way to get to escape from PFAS, but also to use those fluorinated group as a limb chain. And for those, of course, you will have to design original reagent and transformation. I think it's very interesting to consider a successful partnership of homogeneous catalysis and organofluorine chemistry, and also to valorize small fluorinated building blocks as feedstock for complex molecule synthesis. So the first part of my talk will be dedicated to the valorization of valuable fluorinated building blocks. Indeed, we have been interested in BTP, which is a 2-bromo-3,3,3-trifluoropropene. Why is that? It's a commercially available, easy to handle, and stable reagent. Is a liquid, at least in Normandy, in France, and it's more importantly, it's very inexpensive. So here you have a chance to take some waste from industry and to be able to use it as a building block for valorizing again this molecule in something in compound of more and broad interest. Indeed, if you look at the price, I mean, it's less €2 per gram, which is highly attractive if you consider how costly fluorinated reagents might be. It was even considered as an interesting candidate for use as a fire suppressant. And of course, a lot of team have been already focusing on how to use it in cross-coupling reaction. So we were interested to this moiety because if you look at that trifluoromethylated olefin, you can find this moiety in various compounds, such as trifluoridine or bifenthrin. But how could we got access to a large library of compounds? The first thing you can think of is to take benefits of the presence of the bromine atom and to envision a carbon-bromine bond cleavage. And this is something we investigated while using a directing group to ensure the radioselectivity in transition metal-catalyzed transformation. But then we were thinking of, could we also selectively convert this CH bond into a carbon functional group bond? And this is what I would like to develop today. So if you look at the synthesis of beta-trifluoromethylstyrene, there are a lot of possibilities to get access to those compounds. You might imagine trifluoromethylation of styrene derivative, hydrotrifluoromethylation of alkynes. You can also build up olefin or use those CF3-containing building blocks in cross-coupling reactions. But what about getting an access to beta-trifluoromethylstyrene by the transition metal-catalyzed CH bond activation? And this is where we started our study. If you look at, let's say, an ideal scenario will be to start from a non-prefunctionalized substrate bearing a CH bond and a directing group to ensure the radioselectivity, that you might couple it in a CDC coupling pathway with one of those reagents. And here you may have an access to the targeted molecules in H2 as a byproduct. A close look to this synthetic pathway quickly make us realizing that this is going to be quite difficult to get this reaction running in lab, since the boiling point of those two reagents is pretty low. So you might need a special setup. So then we thought maybe we can find a solution from that and use BTP as a coupling partner. Why is that? Under basic condition, you might generate this trifluoromethylated alkyne, and then because for instance, INU in 2014 has shown that under basic condition, you can generate it and then undergo several reactions. And this is one of the numerous examples you might find. And from this in situ generated CF3-containing building block, then you might envision using ruthenium, which is known for hydroxylation reaction, to cross those two reagents to get the expected compound. And indeed, the reaction was working pretty fine. We were able to functionalize a large panel of 2-phenylpyridine derivative, noting that we need this methyl on that position to avoid any difunctionalization. And you can see that the reaction was broadly applicable with electron donating group, nitro, CF3. We were able to confirm also the radioselectivity and the stereoselectivity and the E-olefin formation thanks to this X-ray structure. And you can see that you can also functionalize inversely substituted as a para, meta, orthofunctionalized derivatives, as well as heteroaromatic derivatives. However, you can also see that depending on the nature of the functional group, especially this substituent NH-Boc or this cyano, the yield were not so synthetically interesting. But I will come back to this later on. So from this, we wanted to understand how that works. And one thing we realize is we can prepare from two ruthenium complexes. And in both cases, you might replace the ruthenium precursor as either the complex A and complex B. And in both cases, you will get expected compound. So the take-home message here is this ruthenase cycle is a key intermediate in the process. But interestingly, we noticed that we can even increase the yield by using this complex A. So not only we don't need this paracymene as a ligand on the ruthenium metal, but also we can maybe tune the reactivity using directly this ruthenase cycle as a catalyst in the transformation. So intriguing about this, we ran again a couple of experiments, and what we found out, if you compare the standard condition here in gray with the condition using this complex A as so this metal cycle as a catalyst, you will see that we get even better yield or similar yield. We can even increase it in the case of NH-Bock. But more interestingly, you can see that in the case of the cyano, we can now reach 78% for the formation of the desired product compared to 44% using the commercially available precursor. So then we conducted further mechanistic studies, and we showed that the biokinetic isotopic effect, that the CH activation step might be the redetermining one. And then we thought, we have learned that this metacycle is a key intermediate and a key catalyst in this transformation. Thus, this reaction then might undergo via the generation of bis cycle complexes. So this concept have been pioneered by La Rosa and Ackermann using ruthenium-2, ruthenium-4 process. And in our case, since we are working in a redox neutral ruthenium system with this hydroarylation process, we were wondering if it was possible to imagine this. And indeed, to confirm that, we first conducted a stoichiometric experiment with this complex A in presence of BTP. And what we can notice, if you have no additive, you have only barely on traces of the compound. But if you add the substrate, suddenly the reactivity is back. So then we thought maybe this pyridine might be just playing the role of ligand. And that's why we added pyridine, and bipyridine is a reaction mixture. But you can see that we can get lower yield than the one we observed with 1A. We strongly support the formation of bis metacycle. To further have confirmation of this bis cycle complex formation, then we conducted another reaction. So starting with a stoichiometric amount of this complex A in presence of BTP and running the reaction for 2 hours on a basic condition, what we observe is almost no reaction. And as soon as you had the substrate, after 2 hours, you can see that now the reactivity is back and we can get descended towards the desired compound. We also conducted some GCFID experiment to further confirm the liberation of this paracymene. And as expected, when you have consumption of this starting material, you will see in the meantime the release of the paracymene. But what was very interesting here is, as you can see, there is an incubation time necessary to the formation of the product, which might be another clue towards the formation of this bis cycle complex. So with all of this data and supporting by DFT calculation, we were able to propose this following mechanism. So first of all, this substrate will coordinate to the ruthenium precatalyst and then form this intermediate A, which is an active catalyst in our system, which then might react with this species, this CF3 species generated in situ in the course of the reaction, leading to this molecule. Then this intermediate one will be able to generate this intermediate 2 thanks to the generation of this bis cycle intermediate. And then after coordination of DMF, and we will see later on how those additive might be crucial in the coordination sphere of the metal, we can get this species 3, which then might undergo carboretination, and upon the presence of another molecule of substrate will generate the product and regenerate the species 1, which is active in the catalytic cycle. So the second part of my talk will be more dedicated about emergent fluorinated group. So the first, when we jump in that field, the first interest we have was about SCF3 group. Indeed, the SCF3 group is a very interesting fluorinated moiety. It has high lipophilicity, and you can see that the lipophilicity of SCF3 is even higher than the one used with CF3-1 and have a very strong electron withdrawing character. And interestingly, you can see that this moiety already finds suitable application in various drugs and agrochemicals. So here, I would like to stress out the fipronil, which is an insecticide supplied by BASF, or tetraxuril, which is supplied by Bayer. But I also wanted to say that there is also a strong interest about pharmaceutical company, and especially with AbbVie, for which this molecule is a key intermediate in the navitoclax synthesis, which is a blood cancer candidate in phase 2. So over the years, we have been mainly interested in how to introduce this SCF3 moiety by the direct functionalization of a CH bond. And we have been working quite a lot with two electron manifold. And more recently, we were wondering, could we then now use this SCA-free reagent via one electron manifold and maybe discover novel reactivity? And this is how we turn out to the formation this fluorinated acyclic thioaminyl derivatives. So indeed, if you look at what has been developed in the CH functionalization at the alpha position of an amide by photocatalysis, this is quite a recent interest, since there are over 10 publications, 8 were reported after 2016. We were mainly focusing on the carbon-carbon bond formation with a single example of carbon-oxygen bond formation. So with this in mind, we were wondering, what about the possibility to create a carbon-sulfur bond? So when we started this optimization, we use a Munavali reagent, which is a well-known reagent for SCF resources. And it turned out with using these organic dyes, we were able to functionalize a large panel of benzamide derivative with halogen, ester, but also heteroaromatic derivatives. It was not restricted to aromatic benzamide, since we can also have these phenylcyclopropyl acid derivatives, this terbutyl-1, the adamantal, but also this camphoronic acid derivatives. Interestingly, when you have two possible positions to be functionalized, such as this benzylic position of this methyl-1, you can see that we have exclusively the functionalization of the targeted position with the SCF3. So we were curious to know about how this reaction was working. So we conducted some mechanistic studies. And the first thing we do was the kinetic studies. And at the beginning, when we mix the starting material with this Munavali reagent, you can see the formation of the expated compound and the disulfide, which is a well-known byproduct, I would say, when you are working with SCF-free compounds. But what we were not anticipating when we start looking at this transformation is a key role of this SCF-free dimer. Indeed, a close look to the kinetic studies clearly indicate you that while the Munavali reagent is consumed over time during the reaction, your first species you will generate in situ in the reaction mixture is this SCF-free dimer here in the red, and after only an incubation time, you will see the formation of the expated product. Plus, you can see that after 2 hours, it seems that the product was not so stable. So the first thing we wanted to understand is it a degradation pathway or not? And indeed, we confirm that the stability of the product under open irradiation might not be so high, since degradation, some part of the product was degraded. Then we conducted some Stelbomer fluorescence quenching analysis to understand a little bit better how that works. And we clearly identified that this Munavali reagent was quenching the excited state of the photocatalyst. Cyclovoltammetry experiment also showed that this Munavali reagent might react with an oxidative quenching might be involved to using this photocatalyst. While if you look at the CV of the benzamide, no oxidation are possible considering the redox potential of this photocatalyst. So with this in mind, we were wondering, how is it possible? We know that the reaction is working, but the data we got was quite counterintuitive with what we observed. But remember, the first thing that happened in this reaction is the generation of this dimer. But if you liberate this dimer in the course of the reaction, starting from this Munavali reagent, you will also liberate the corresponding phthalimide anion. And indeed, when we added some phthalimide anion on the reaction mixture and conducted some new CV, you can see that the redox potential now is much more affordable. It's reaching 1.43 volt. So with this data, we thought that the PCAT process might be involved in this transformation. And this was also further confirmed by kinetic isotopic effect. We also conducted some control experiment with radical scavengers, indicating that the radical process is likely to be involved in the transformation. We reached a quantum mean of 0.59, which strongly support that or indicate that there is no radical chain process. And finally, we identified the key role of this SCF3 dimer. Indeed, starting from this silver SCF3, if you mix it in THF with NCS for 2 hours, you can get exclusively this dimer, which is very typical with this chemical shift of minus 46.1. And then using similar conditions, so adding the starting materials, organic dyes, and this mixture of solvent, we were able to get 68% of the desired product. Just to make sure about how the influence of the mixture of solvent might impact the outcome of the reaction, we also run this control experiment, which strongly support that the dimer of CF3 is a key active species in this process. So with all of this data, we were able to propose this mechanism. So photoexcitation of this photocatalyst, and then we will have like oxidative quenching of this Moonavary reagent, liberating this SCF3 radical, which might rebound to the formation of this dimer with the generation of this styrimidine anion. Then the oxidized photocatalyst will then undergo a PCAT process and liberating after a 1,2-H transfer to this intermediate free, and this later will then be able to react with this SCF3 dimer towards the expated compound. So the SCF3 was the starting point of our journey in emergent fluorinated group. We also have a strong interest in having new emergent fluorinated moieties. And especially, I mean, our group and others in the world were very proactive in that direction. I mean, having this SCF2H or having a functional group like, for instance, a ketone, a sulfone, an ester might give a chance to modulate at will the properties of molecule in which those fluorinated reagent might be incorporated with. So, of course, there was a craze and a big interest in the development of indirect and direct methodologies, and we jump in that field to see if we can push forward what is known. So we recently worked on this SCF2 sulfone, which is known to be the chameleon fluorinated reagent. As you see, it might be easily converted in various fluorinated moieties, CF2H with double bond CF2 or with CF2R, which are key fluorinated residue in various compounds such as eflornithine or selectracetam. Of course, there were already brilliant work from Wu and Shibata's group, in which they were showing electrophilic sources such as this iodonium derivative or this sulfonium 1. However, when you look at the application, in the case of the Wu reagent, you can easily make a sulfur CF2 sulfone bond or undergo copper-catalyzed decarboxylative difluoromethylation reaction starting from the corresponding vinylic carboxylic derivative or the allylic one. And in case of the Shibata reagent, it was very efficient for the formation of a carbon CF3 as CF2 sulfone. But what about having a reagent which can combine both reactivity and go even further? And this is exactly what we were looking for. We wanted to design a bench-stable metal-free synthesis and using those non-depleting reagents in the course of the synthesis of these electrophilic sources. And inspired, thanks to the knowledge we have of the Yumimoto group, we know that the nature of this skeleton might be the key to tune both reactivity and stability. So we undergo the synthesis of this reagent. So starting from this dimer, we can easily get this SCF2 sulfone derivatives. Then selective oxidation in presence of MCPBA will lead to this sulfoxide, which upon treatment with trifluoric TF2O will lead to the expated compound. And here, I would like to say that Enzo was involved in this project, was very efficient, since over this 3 step, you have only one purification step after this oxidation, and we can get a gram scale synthesis, since more than 40 grams of the reagent was possible to obtain in a 1-run process. And on top of that, this reagent is air and moisture tolerant. So we wanted to showcase how efficient is this reagent, and to do so, we were able to show that we were able not only to make carbon sp3 CF2 sulfone or sulfur CF2 sulfone derivative, in similar yield as the one obtained in the case of the Jimbo-Hu or Shibata's reagent, but we were also able to further diversify the class of compound we can functionalize, such as these aniline derivatives, electron-rich heteroatom, and electron-rich heterocyclic compounds. Then we were wondering, fine, we have a very efficient electrophilic sources, but what about functionalize a simple carbon H bond? And indeed, what has been reported a couple of years ago, and period where were reported by Lowe, followed by us, we were able to promote to develop a copper catalyzer mediated trifluoromethylation of this carbon CF3. But so far, there was only 2 reports. So what about our working hypothesis? It would be, would it be possible to make a carbon CF2 functional group starting from this carbon H bond? So ideally, we would like to use a native functional group, avoiding multi-step synthesis for the introduction of the derating group and eventually its cleavage, while keeping the same and high Z selectivity for the final product. And ideally, we can use non-noble transition method. That would be fantastic. But don't forget that moving from a CF3 to a CF2 functional group completely changed the reactivity of the whole system, since this CF2 functional group is itself quite reactive due to the presence of the functional group and prone to undergo side reactions such as difluorocarbene generation of radical CF2 residue. So we first developed a catalytic process, and you can see that we were able to functionalize a large variety of alpha aryl acrylamide derivative, but also one substituted with an heteroaryl alkyl derivative as well, but also beta and alpha beta disubstituted acrylamide derivatives. The key for this was to use a copper catalyst and also combined with a silver fluoride, silver BF4 salt in the presence of NMF and TFA. And the reaction was quite robust since we can conduct it under air at 80 degrees. But still, having this copper catalysis was, from a fundamental point of view, very important for us because there are not so many examples of copper-catalyzed CH1 functionalization. But we thought that if we want to push forward and if we want people to use this methodology, and especially industry, I think mixing copper and silver is not a great idea because the price of the process is too high. So then we also developed a copper-promoted transformation. And as you can see, we got similar or even similar or yielded the same range using this methodology. So we were very pleased about that. So then we wanted to understand how it works. And especially, why do we need NMF and TFA? So first, we try to replace NMF, and you can see that we can use other additives, but NMF is still the most efficient additive in this transformation. And thanks to a productive and fruitful collaboration with Professor Jamal Moussaf from Emory University, we were able then to understand a little bit better how this process is working. And the lesson we learned from that was that the copper-2, copper-2, copper-3, copper-1 disproportionation promoted by TFA was involved in this transformation. We also confirmed that the CH activation was a redetermining step, both experimentally but also computationally. And finally, we found out that NMF was playing main role in this transformation in the CH1 activation step, ligand exchange, and CC coupling. So with all of this big picture in mind, we were able then to provide a new mechanism. So first, ligand exchange leading to this copper-2 NMF2, and then thanks to this copper-2 copper-2 disproportionation, we can get the expected intermediate A, which then might undergo to the formation of this metal cycle, which is a redetermining step and irreversible. And finally, we were able, thanks to ligand exchange in the presence of the electrophilic sources, to get the expected intermediate C. And after CC coupling formation, we can get the expected compound, while upon the presence of an oxidant, you can regenerate the copper-1 to copper-2. Now we'd like to switch a little bit more to another part of my research interest, which is more related to how could we functionalize CH bond, and especially how could we develop strategies to access polyfunctionalized arene. So I think I don't have to convince you to tell when I'm telling you that polysubstituted arene are found everywhere in pharmaceutical materials and agrochemicals. So more specifically, we were particularly interesting in this amide, in which you have a substituent at the ortho position and halogen on this position. Indeed, if you look at a closer look to this moiety, you can find it in various compounds of interest, such as this anti-diabetic or this anti-sickness, which are widely used. And in all cases, you can find a similar pattern, a methoxy group and a halogen, chlorine, bromine. So with our knowledge about CH bond activation, we thought that maybe we can undergo sequential functionalization and start from a non-prefunctionalized substrate and selectively introduce two functional groups in a very selective manner. While the ortho prime difunctionalization was well studied, the ortho meta or ortho para was not were still underexplored at the time we started this project, and still there are a lot of things to do. So we come with this idea that maybe we can undergo ortho CH, meta CH difunctionalization, starting from this secondary amide with a native functional group to get access to the desired compound. Of course, the key challenge here will be the complete selectivity to have this functional group one here at the ortho position, while nucleophile at this meta position compared to the amide, but also the compatibility of all reagents between the first step and the second step. And if we can use inexpensive coupling partners such as methanol or sodium halide, that will be a fantastic point. And indeed, this is something we were able to conduct. So starting from this methoxylation, we thought that we can undergo this methoxylation by CH bond activation, and then in the presence of sodium halide and oxidant, we might favor taking benefit from this or methoxy group, a metal-free oxidative para-halogenation towards expected compound. And indeed, the reaction was working pretty fine in a one-part process and a sequential one. And then we were able to functionalize a large panel of compounds. So I will not go into details, but the key point here is you can play and dance with all the halogen. You may have the iodine here and the bromine here. You can have a chlorine, the bromine, the fluorine, the bromine. So if you think about how could you synthesize those compounds in a very selective manner, this is not so true. This is not so easy. Then we were able, in this case, via a two-step process to introduce the TT, which is a tantalum salt. And we know, thanks to the work of Ritter, that among others, that we can functionalize this tantalum salt in various manners to get a library of compounds. And finally, we consider that we reach a platform, a synthetic platform, since this might be derived into the corresponding carboxylic acid, alcohol. You can even functionalize it, but you can also take benefit of the presence of this halogen with and engage it in a various coupling reaction and to further functionalize this molecule. So finally, we showed that we were able also to get an access to a methicillin, that with this approach, so using our standard condition, you can have an access to this key intermediate. Then methylation of the derating group and saponification will lead to this key carboxylic acid, and upon classical transformation toward the formation of the corresponding amide, we can get an access to methylglutinide, which is an diabetic drug, as mentioned earlier. So we also have a strong interest in how could we escape from derating groups. So already I show you that we have a possibility to use the native functional group, and a lot of groups have been particularly involved in that research, using carboxylic acid or even starting from chlorobenzene as a solvent. And the key challenge there are, of course, reactivity and reagent selectivity. And here I would like to stress out the work of a couple of key players in the field. But what we then were particularly interesting again is, could we converge our interest in the synthesis of difunctionalized arene while having no derating group? So this difunctionalization, and I think the most famous way to reach those and efficient way, is the Catania reaction, which is well established, and there are a lot of major and tremendous advances made over the years. And with this, we thought that maybe we can also play with this and combine our interest, starting from a simple molecule with two CH bonds, and be able to selectively introduce one functional group and another one with control with high reactivity and the total control of radioselectivity. So to do so, we imagine that we may have the need to use a traceless leaving group. And this is exactly what those talented students were helping me in this project, thanks to the synthesis of 1,2,3, 1,2,3,4, and 1,3,4 substituted derivatives. The idea was to use tantrination. I mentioned already the work from Ritter, who showed that the tantrination might be even more selective than the halogenation. So we thought that maybe we can start from non-prefunctionalized substrate, use tantrination strategy to get this molecule, and then use tantrinium as a traceless directing group, and then undergo a catalytic tap reaction to get the expected compound. And so far, the key challenge was here, could we use tantalum and sulfonium salts more generally in a catalytic tap reaction, which was not so clear at the beginning. And this is how we designed the reaction. So first of all, we synthesized with tantrin S oxide, and upon condition, suitable catalytic condition, using this NBE as a transient mediator, this ligand, and this phosphine, we were then able to functionalize a panel of derivatives. And here I would like to stress out that we were able to functionalize challenging 1, 2, 3, 4 aromatic derivatives, and then able to basically modify any coupling partner of this transformation. Because we wanted to show that these tantrinium approaches, which is an alternative now to this catalytic tap reaction starting from halogen derivative, we were interested to see how far we can go, and then we were able to undergo the generation of this dealkylated compounds with the total radioselectivity thanks to ipso working chain process. We were able also to undergo an orthoarylation, ipso olefination, as well as orthoalkylation, ipso CHR and orthoalkylation, ipso cyanation. And finally, we were able to develop a small scope in which now we are able to make orthoamination, ipso-olefination, and orthotiolation, ipso-olefination. So with this, we have a full tool set to get selective access to dye-substituted compounds. And finally, I would like to talk about the new strategies we have also envisioned to develop and directed this Distal C-H functionalization. Indeed, since the beginning of my talk, I've mainly focused on either proximal functionalization, such as orthofunctionalization or orthometa functionalization, thanks to the difunctionalization strategy. But what about if we can start from a native functional group and be able to selectively address one position to another on aliphatic derivatives? So again, we thought that we can take benefit from this emergent fluorinated group, and especially we wanted to introduce them on derivatives which have a functional group such as alcohol. So we first take benefit from the library of compounds which were of reagents which were available, especially thanks to the group of Kilong Chen, but also our own contribution. And then we were able to the idea was to be able to start from an alcohol. No need to pre-activate it to get the expected compound with a total radioselectivity. So inspired by the major work from the group of Zhu, we thought that we might use PIDA and generate in situ this intermediate after ligand exchange between the acetate and this alcohol. And then upon irradiation, the alkoxy radical might be generated, followed by 1,5 hat towards the formation of this intermediate 3, which then might react with SCF2 reagent or the in situ dimer. And indeed, the reaction was working pretty well. We were able to functionalize a large panel of alcohol. And as you can see, it's working with unprotected primary, secondary, and tertiary alcohol with a total radioselectivity thanks to this metal-free process to the delta functionalization. And we were able to not only introduce the SCF-free moiety, but also SCF2 ester, SCF2 phosphonate, and to some extent, SCF2H group. So with this, I hope I show you a little bit about our interest about proximal and distal C-H functionalization and the different strategy we may have, and also our strong interest in organofluorine chemistry. And I'm very happy to tell you that this reagent is now available in ABCR. Of course, I have to thank all the past and current members of my group with who I will never be there today. I mean, they are doing an amazing job and I'm very grateful. I would like also to thank all the suppliers for fundings and of course, all of you for your kind attention and being joining me for this great event. And thank you, Neha, again for this kind invitation.
Thank you so much, Professor Besset. It was a wonderful talk.